Welcome back to our class. Today we're gonna be looking at the paper 2023 final exam. Yeah, so share the video so that everyone is here. We can discuss this paper. No one should miss out. This paper was really a very easy paper for those people who did the study, and for those people who did use my book for distinction material. Yeah, if you want it, you always get it from the link below. So this paper was a little bit easy, was just a little bit of easy, and we're gonna see how we can really see that those people, they can mark themselves and then they see, and also those who want to prepare for the next exam. All right, let's start without wasting time. M.SAIDI, as usual. According to Lamarck's theory, I know most of you don't like Lamarck because of his theory, but me, I like him because he started the story. All right, according to Lamarck's theory of evolution, or for evolution, acquired characteristics are not inherited. Obviously, we say that Lamarck says that acquired characteristics are inherited to the next generation. If you have a person, maybe your father gets an accident or is a bodybuilder, and then you're going to produce kids who are having muscles already. Acquired characteristic is inherited. So here the key point is not. So you have to take note of that. So this one was off. And then they are saying that organisms evolve because they don't because they adapted to their environment. No, here they are saying that organisms evolve because of the adaptation to the environment. How do organisms evolve due to the adaptation of the environment? Is it natural selection? Because adaptation will require that those organisms which are able to adapt will evolve, and those organisms which cannot adapt will die. Yes. Then number 3, they're saying that there is variation among organisms. Lamarck doesn't talk about variation. Yes, so this one is off. Then they're saying environmental change has no influence on the species diversity. Here, they're saying that environmental change has no influence on the diversity of the species. And then here they're telling you that organisms evolve because they adapt to their environment. So how are these two different? Here it means that environmental change have no influence. For example, that if there is scarcity of food, yes, and then the one which is on ground die out. This is opposite of this, né? And they die out, and then there will be no influence on the species, how they diverse each they are different from each other. So that is wrong, because species will change based on the what they want, but still based on the environment. If they want to be, for example, they want to stretch their necks, they stretch their necks until they reach, for example, the tree. If we are using the giraffe, yeah, you call them giraffe, yes, because that's the best example most of the people are looking at. So the best answer here is gonna be B. And then they're saying that genotype for species characteristics. When you talk about the genotype for species characteristics, what does it mean? They're saying that contain 2 chromosomes. No. Physical appearance, they're saying genotype. Physical appearance is a phenotype. No. Is the composition of a gene pair. Look, they're not saying that genes. They're saying gene pair. Meaning that you have, remember that when you talk about an allele, is also a gene, but is half of a gene. or alternative form in which a gene can exist. For example, tallness is an allele, and also shortness is an allele. For example, capital T, small t, for example. Yes, so this is an allele, this is an allele, but this is a gene, meaning that it's a pair which makes a what? A gene. And then is represented by the genotype of a specific characteristic. Here, characteristic. Here they're saying it is represented by one allele. It's not represented by one allele. Even if it is shortness, it will be represented by the two alleles. Yes, for example, capital R, or small t, they are saying a characteristic. Yes, so when you talk about the characteristic, it means that they are looking for the complete set of that characteristic. So the best answer here becomes C. And then they're saying after discovery of fossil, scientists classify it as after discovery of a fossil, scientists classified it as an African ape because it had, okay, what do African apes have? Large cranium? No. Small cranium? Yes, they have small cranium. Small cranium? Yes. Large cranium? No. So meaning that A and D are out. So these two are out. So let's look at this. Small cranium, non-prognanthus, meaning that they don't have a protruding jaw. Apes have protruding jaws. Yes, prognanthus means protruding jaws. So prognanthus, this is correct, this is wrong. So it means that the best answer is C. They're saying that large canine. This one is correct, this one is correct, but this is wrong. Therefore, B is wiped out. The best answer is C. So C is going to be the most correct answer. Yes, let's go to 1.1.4. The statement below describes the step in the process of cloning. I explained this, the process of cloning yesterday very well, but let's ignore that. You are not there. So now let's look at it. Remember, they are saying that embryo, okay, let me just show you here so that number one, you get one cell, you get a cell A and cell B. So this is animal A, animal B. Get a cell, ovum. I'm just summarizing it very fast. Somatic cell. So here is somatic cell, meaning that is 2N. After that, remove the nucleus of the ovum. Yes, throw it away. Take the what? The casing. And then here, remove the nucleus because I want the characteristic of this. Bring the nucleus, throw the casing. So bring the casing here and bring the nucleus, fuse them together with electric shock. Yes, after that, bring these two, once it is fused together, it looks like a zygote-like structure. Therefore, it becomes, I'm just explaining so that because this is the process they are trying to show you, it undergoes mitosis. Bas. Then, let's go back to the question. They're saying that the statement below describe the steps involving in the process of cloning. Embryo in the implanted is implanted into the uterus of an adult female. Embryo is implanted. This must be around this stage 4. Okay, we shall find out. Nucleus from the somatic cell of the donor cell is extracted. This could be 1. Nucleus. Check. Nucleus. They're saying the nucleus or the cell? Nucleus from the somatic cell. That's not step 1. You can't extract the cell before you get the cell. You can't extract the nucleus from the cell before you get the cell. So we need to first get the cell. We are looking for these two steps of getting these two cells. All right, we go back. They're saying that the nucleus from the somatic cell is inserted into the ovum. The nucleus from the ovum of another is removed. The ovum from, okay, now we have to look for it. They're saying that the nucleus from the somatic cell, meaning that, okay, they obtain the cell, yes, but now after obtaining the cell, in this statement, they're not talking about this. So meaning that they're starting from this. They're not talking about this. They're starting from 1, 2. Okay, let's go back. They are saying that the nucleus from the somatic cell of the donor is extracted, meaning that the nucleus is removed, is brought here. So this could be step 1. Okay, we shall see if it is the most correct. Step 1, so this is covered. They are saying that the nucleus from, yes, the nucleus from the somatic cell is inserted into the ovum. The nucleus, this is done. The nucleus is of the somatic cell is inserted into the ovum. Do we have the ovum? The nucleus from the ovum of another individual is removed. This is step 2. Yes, this should be step 2. Because I removed here the nucleus, so I remained with this. So now I inserted this into this. This should be step 3. The nucleus from the ovum of another cell is removed. It's done. The ovum of the new cell is given. The nucleus from the somatic cell is inserted. Now this is step 3. So meaning that now I have 2, 4, 3. Then I'm remaining with this and this. So they are saying that the ovum is now, the ovum with a new nucleus is now given electric shock. Now we are here. We are giving it electric shock, meaning that now this becomes 4. Yes, and then now the embryo is implanted into. So meaning that I'm going to have, yes, it's going to be 2, yes, 4, yes, 3, 4, 1. Meaning that 2, 4, 3, 5, 1. So this is the most correct answer. So the answer is A. Let's go to the next question. Then they are saying that 1.5, shrubs, these are short grasses of the family. This family, they are saying that can be found in Australia, South America, Indochina, and parts of Africa, as shown in the map below. It is hypothesized that all continents were once one large continent called Pangaea, and that they separated due to continental drift. They said, okay, sharp. They're saying that this is evidence. This evidence that the family, yes, this family, what about it? All belong to the same species. No, they don't belong to the same species. They don't belong. That's not an evidence to show that they belong to the same species. Number 2, they're saying that they are equal distributed in all continents. Who told you that? They are not equally distributed. Are you trying to mean that what is here and what is in South America and what is in Africa, they are equal? No. Wrong. They're saying become extinct. They're still existing. Arose from a common ancestor. Simple, because at first they were together. Yes, but they went to different environment due to the separation of the continent. And then they developed into different species. That's why we say that they don't belong to the same species. Therefore, D is the most correct answer. 1.6. Yeah, these ones, they take some time, but when you reach there, we're going to be faster. The diagram below shows the DNA profile of a girl that her mother and 4 males. What about the males? Okay, maybe they are claiming. Which male is a girl's biological father? I have to look for all the bands which correspond to the mother, because the mother is known. This goes with the mother. Done. This goes with the mother. This goes with the mother. Now, the remaining bands from the girl must be matching with the guy who is claiming. Okay, let's go. This one matches. Cool. Yes. This one matches. Is there any? Yeah, because this one is matching. So is there any other for the girl? No. So it means that this is number 3 is the biological father. It must All of them must be ticked for the case of the girl, but they must be the remaining one, which did not match with the mother, must match with the father. Hence, number 3 is the most correct answer, which is C. Okay, the following are the characteristics of group of animals. Yes, animals. If you're saying animals, just a moment, guys. Okay. Just a moment. Okay. Animals. They're saying that which characteristics of group of animal able? Which are the characteristics of group of animals able to interbreed, occupy the same habitat, produce infertile, belong to the same species? Which combination correctly represents a population? Which combination represents a population? Yeah, it means that you can't know which group represents a population. Just a moment, guys. Let me increase here so that I can see clearly. Yes. Yes. Which population does the population is being shown by what? Is being shown by the able to interbreed, and they must produce a fertile offspring, occupy the same habitat. Yes, they must be able to interbreed, but interbreeding they must produce the fertile offspring. And the only word which can help us to bring about to produce a fertile offspring is one word. Which word is that? Same species. They belong to the same species. So the answer is B. Same habitat, same species. Yeah. So we go to the answer becomes 1, 2, 4. 1, 2, 4. Where is 1, 2, 4? Is there any 1, 2, 4? 1. 1, 2, 4. Yeah, it's B. Yes, it's B. So yeah, it gives me the correct answer. Let's go 1.8. Which diagram? Let me see. Which diagram? Not which diagram. During anaphase 2 of meiosis, the two chromatids of the chromosomes are pulled apart, each moving towards the opposite poles at a rate of 1 micrometer per second. This question, okay. I see it as a new content to students. Anyway, but it's just an application question. Okay, they're saying that, yes, two chromatids of the chromosomes are pulled apart, each moving towards opposite poles at a rate, this is the key point, at a rate of 1 micrometer per second. Okay, each second, 1 second, it means a distance moved is 1 micrometer. Let me say 1 like that. The distance in micrometers between the chromatids after 20 seconds. Obviously, if it's 1 second, 1 micrometer, after 20 seconds, they'll have moved 20 micrometers. So that is a direct question. However much is just an application question. So the answer becomes B. Yes. So the answer there becomes B. Yes, let's go to the next question. The next question, they're saying that, which one of the following indicates the type of variation? I gave you examples yesterday. Anything which does not change since you are born is continuous variation. Anything which changes as you are growing up, it's continuous variation. For example, here they gave you height. Height changes, therefore is continuous. The way you are born and the way how you are now is not the same way. Height is continuous. So it's where there is this continuous is, I think I have the answer now. Because the rest are wrong for the height. And it can't be correct for the next one. Okay, skin color. I don't think most of us, we are born yellow bones. Check us now. Hey, we are original indigenous. Yeah. Yes, color changes, skin color changes. You check the kind of people. Some people, I want the black one. I want the light skinned one. I don't know what you're going to use for them for, but it changes among the individual. All right, this is continuous. Check. Earlobes, the way those earlobes are since we are born, unless you get an accident and then they change. But if not, it's the same thing. So, correct. So, let's not waste time there. Answer is B. Let's go to terminologies. Terminologies. I know sometimes the terminologies, they make these students a little bit of a problem with it. But I don't think these terminologies were that hard. Ne? Let's go. Let's go. The first terminology, they are saying the type of inheritance which produce intermediate phenotype. Intermediate phenotype. Don't forget that we said it in the what? In the distinction material book, our distinction material book. Ne? Yeah, for those people who were not there. But okay, there we go. Intermediate. How do you call this? Incomplete. Incomplete dominance. Intermediate. Yes, we talked about it. So the answer here is incomplete. Complete dominancy. Yes, done. The point at which adjacent chromatids overlap during crossing over. Chiasma. Chiasma. Guys, spelling is mandatory. The process by which information is copied from DNA to RNA. Transcription. Told you. Transcription. Let's go to the next question. What are they saying? They're saying that sugar found in DNA. Sugar. Thymine. They're saying sugar, not base. Gone. Thymine. That's a base. They're saying sugar found in DNA. Ribose is found in RNA. So for the DNA, it's going to be deoxy. That's why he's saying D. DNA, deoxyribose nucleic acid. So the sugar found there is deoxyribose. The bond that occur between nitrogenous bases in DNA molecule. The bond that occur between nitrogenous bases in DNA molecule. So how do you call that bond? That bond is weak hydrogen bond. Yes, you must say weak hydrogen bonds. Most of the people, they wrote peptide bond. Peptide bond. In some cultures, they say that sometimes you can laugh because of sadness. Not because you are happy, but you are sad. The same way someone can cry because of happiness. When I laugh at because someone is because of the sadness. So this is weak hydrogen bond, not peptide bond. Yes, because I highlighted it that these are the mistakes students do. Yes, okay, let's go. Manipulation of organism's gene to obtain the desirable characteristic. How do you call that? Manipulation of a gene to get a desirable characteristic. Whenever you talk about manipulating in science, whenever you say manipulating, what did I say? I said that is engineering. Engineering. Manipulating. But you are manipulating genes. So, what are you manipulating? Genes. Therefore, genetic. Simple. Genetic engineering. Guys, I told you don't need to cram. You don't need to cram. You just understand life science. Tell the people to join the channel. If you want a distinction, Sanda a Duke. Yeah. Use the distinction material. There is no way how you can fail a paper. Never. Yes. Okay. Unless you don't want to. All right. Unless you found me. You found the channel late. But if you want a distinction, tell the whole world. Thunder Duke. Okay. They're saying that representation showing the arrangement of diploid set of chromosomes. Hey. Representation. They are looking for how do we call that representation? Which shows the diploid set of chromosomes, meaning that chromosome number 1, chromosome number 2, chromosome number, but they are saying, see, diploid. Like that, we call it a karyotype. Karyotype. Done. Terminologies out of 7, 7. Okay, source of variation. How do you call random? We talked about this yesterday. Random mating, random fertilization. Source of variation for the species. I told you that a man produces over 500 million sperms. That is a rich man. That's why they tell you that you are 1 in a million. It means that your sperm was 1 in a million sperms which were released by your father and 2 there, and then only 1 you fertilize the ovum. So random fertilization is also random mating. You don't just find people come and say that, okay, now I want this one. You just go and then marry. You just say, I want the small one, I want the big one, I want the light skin, I want the dark skin. You see, you're not doing it randomly. So you are minimizing variation. So variation, it must be random mating. When you are chasing this one and then another one comes into your eyes, you just go with that random mating, like a chicken. Yes. So this is both A and B. The answer is not A and B. The answer is both A and B. I explained it yesterday. Both A and B. Organisms that possess 2 factors which separate so that each gamete contain only one of these factors. Law of dominance. There is no domination here. Law of independent assortment. Organisms possess 2 factors that which separate that each gamete. Law of independent assortment. This is done in dihybrid cross. We call this law of segregation. Law of segregation, not principle of independence. So the answer here is none. Because this is a law of segregation by Mendel. You remember that guy Mendel discovered the law of segregation in 19. He was there in 1865. Yes, Mendel, Georg Mendel, that priest. Yes, Georg Mendel. Yes. His life science is nice. So what you should do is be nice to it. Once you are nice to it, the subject is gonna be nice to you. If you are not nice to it, forget about. Okay, guys, just 1 second so that yes, Mr. Ramaphosa has given back electricity to us, so now we are happy. Yes, it's back. Yeah. All right. So if you see some disconnection a bit, just know that you're just switching the system. But we are continuing. Reproductive isolation. Reproductive isolation. Is my mouse. Okay. Reproductive isolating mechanism in species. You separate organisms through reproductive isolation, breeding in different seasons of the year. Correct. Infertile offspring. This is not correct. This is not correct, Baba. Look, same. This word, these people are confusing us, ne? Breeding in the same season of, no. It's supposed to be breeding at different seasons of the year. Let me show you what these people are trying to trick you up, ne? Let me just show you using the distinction material so that you see exactly how these people are trying to this is a real fake. They are faking us, so that we are off. We are sober. All right. Let me show you what they are showing you. Breeding, yeah, is here. You see, breeding at different times, not breeding at the same. So that answer is wrong. So this answer is wrong. You see, when you're saying the same, the answer is supposed to be breeding. This distinction material, there is no way you can go astray. Yes, so this is wrong. Infertile offspring. Correct. Check here in the book. Check here. Infertile of the way it is without even changing anything. Yes, infertile offspring. So you see, we say that mechanism of reproductive isolation. Now there are some people saying that I wish I knew this booklet. Sorry. Yeah. But you can refer to someone else so that you can get rewards. All right. So the answer here is B only. Yeah. Let's go to the next question. What does it say? They're saying that this is a phase. Ne? What I've seen in this paper as I'm going, I don't know that much what is down, but what I've seen it here is they are using small words to take you off. If you don't read these questions very well, they're just using very small words just to take you off. All right, let's go. So we have to be keen, guys, when we are trying to answer. Yeah. So even me have to read too much so that I don't go astray. I might go astray and give you a wrong answer. No, man. All right, let's go. They're saying that the diagram below represent a cell in early stage. You see? You see these people? The word early. That word, there is early and late. Late stage, mid stage, early stage. So now they're saying early stage. Okay, so if you look at this, which stage is this? This is not interphase. This is early prophase. Chromosomes which are homologous, they align together. Homologous. Yes, crossing over is not yet there, but they have aligned together. And you see that these dots shows that the nuclear membrane is starting to disappear. The nucleolus is already disappeared. You see? These people. So this is prophase. Prophase. Which prophase? Prophase 1. If you don't say prophase 1 is 0. Number of chromatids. So how many chromosomes do you have? 1, 2, 3, 4, 5, 6. So you have 6 chromosomes, but each chromosome has 2 chromatids. So 6 times 2, which is 12. So the answer here is going to be 12. Number of homologous pairs. They are saying number of homologous pairs. They're not saying number of chromosomes. Pairs. So it means that 1 pair, 2 pairs, 3 pairs. 3 pairs. Someone is going to write 6. Ask you, do you have eyes? Yeah. Do you see 6 there? No. I see 3. Why did you write 6? I don't know. Okay, let's go to the next question. Identify what is R. Yeah. R is nuclear membrane. Nuclear membrane. What is S? S is cell membrane. Membrane. Yeah, you know, I told you last time that you guys, you confuse nuclear membrane and the cell membrane. T. What is T? What is now T? T is nucleolus. Nucleoplasm, not in nucleolus. Nucleoplasm. So, the nuclear, okay, maybe they will slash it with the nucleus. Nucleus, yes. But based on this is nucleoplasm or nucleus. So if you give nucleoplasm, yeah, they might give you. If you give nucleus, they might give you. Based on what they have shown us. Ne? Yeah, don't say that. No, me, I wrote nucleus. Yeah, you might be right. Ne? Yeah, correct. All right, let's go. 2 organelles in animal where meiosis takes place. They're asking your parts. The most important structure on you. Which structure is that? Testes. Testes. That's 1. Oh, if you wrote it like this is also correct. Because this is singular, this is plural. Okay? I'm not saying that you write 2. No, just write 1. Hey, students. That's number 1. Number 2 is ovary. So they give you a tick here, they give you a tick there. Simple. They're saying that, you know, they write these funny words of these plants. Okay. Who knows this? I don't know. Let me just quote. The plant carries edible fruit. Scientists have been studying the inheritance of these 2 genes. One for the stem texture, and then another one is for the fruit shape. Okay, sharp. Let's go. They're saying stem can be okay, smooth. Smooth is represented by a capital letter. And then prinkly means that they have those yes, small letter. And then the shape round is capital letter, elongated is small letter. The type of cross, dihybrid. Dihybrid. Dihybrid cross. They're saying state the dominant characteristic in the stem texture. Texture is here. Smooth. Answer here is smooth. Smooth. Boom. Done. Let's go. Recessive. Recessive for what? Fruit shape. Fruit shape, fruit shape, fruit shape, fruit shape. Elongated. Elongated. Let's go. 1.5.3. Give there. The genotype. The genotype. It means that you are supposed to write in terms of gene. Prinkly, stem, and elongated. Prinkly is small letter, so I can't be with the heterozygous. Small n, and then elongated. Elongated is small r. Listen carefully. Listen carefully. If you wrote NNRR is wrong, ask me what? Why? Because of this space. I explained it yesterday. Yes, a space must not be there because this is a genotype. These are not spams. It's a genotype. So it's supposed to be 1. Yes. They're saying that phenotype of plant with capital N is what? Because the small n would be masked. Capital N is smooth, and what? Capital R is what? Round, smooth, and round. Sharp. If you have the space there, is cool, but the genotype is supposed to be no space. Yeah, let me know if you have questions. If you have questions, if you have challenges, yes, I'll smash them. Yeah, I'll smash them. Yes. Okay, let's go. Let's continue to the next question. Which question? All right. They're saying that the table below shows the information selected on hominid fossils. Okay. All right. We are hearing. What are they saying? What are they telling us? They're saying that P. What is P now? P. Oh, the common name. Australopithecus sediba carabo. We say this is a transitional fossil. Missing link between Homo habilis and the. Okay. Who discovered did a bagger? I don't know why they like carabo too much. They brought it in 2017. Yeah. It was the second or third objective. Yeah. Is read the bagger. Lee. Beggar. Yeah. Like that. This is Sediba, Carabo. Yeah, you can and you can find that someone called Carabo is feeding this Carabo. All right. They're saying that Taum child. Oh, Taum, if you're coming from Northwest. Yes. How do we call? Remember, I told you don't go the paper without knowing the 3 Australopithecus africanus species. I said Taum child or Taum child, little foot, Mrs. Place. What are they looking for here? They're saying this is Australopithecus africanus. Cool. Just saying A africanus, they would have given a tick. Yes, because there is no Adipithecus here. So tick A africanus. But they have already given you the spelling here, so there is no stress there. They're saying that who discovered? Who discovered the Taum child? Raymond Dat. Yeah, it's Raymond Dat. Raymond. I don't know whether you call it Raymond or Raymond. Raymond. Dat. Raymond Dat. Check that booklet, the distinction material. Check this distinction material. I organized it nicely for you guys. And even I created a video. Check. Australopithecus, Carabo, read the bagger, Malapa. And then they're looking for the 3 Australopithecus africanus species. Check. Raymond Dat. Taum. Sharp. Let's go. They're saying, name the fossil. P. We have said it. P is Carabo. Then they're saying, identify species R. We said Australopithecus africanus. Let's go. They're saying that, give the name of the scientist who discovered Q. We say that, read the bagger. Yes. Well, you can also give that one brief Elof. Yes, Elof. Wheezy. Yeah, this one. So it depends on whom did you give. Yes. Then I think S, we say that it's Raymond that. Now, by this time, you have 50 marks out of 150. This one is enough for you to shift from a failure, meaning that you're above 30 now, 30%. However, it's not enough for my students like you. Like you. Guys, don't have too much pressure. Like, now I'm failing. That's life. Still, doesn't mean that if you fail this, then life has stopped. No. All right, let's continue. It just make you to prepare, and I pray for you guys so that you pass this, so that you get enough chelete. All right, let's go. They're saying that X, what is X? That is amino acid. I told you guys that you must always label these things before you start. Amino acid. Simple. Let's go. X, Y. What is Y? Y is anticodon. Z, messenger RNA. Let's go. X, amino acid. Z, messenger RNA. Give the nitrogenous base of sequence, nitrogenous base sequence of DNA base triplet. Complementary to complementary DNA base triplet, complementary to mid codon of Z. Oh, they're saying DNA, meaning that we have to convert it backward. Let's say this is 3, this is 3, this is 3. So we are looking for this. So, but we are converting it backward. You have A, U, G. So we're going to be, say A goes with T. U goes with A. G goes with C. So this is the answer. That's what they are looking for. Because they're saying mid in the middle one and for DNA. Then they're saying that give the nitrogenous base of Y. Where is Y now? Oh, the anticodon. So meaning that they want you to change this into transfer RNA, meaning that A goes with the, remember A transfer is an RNA. RNA doesn't have T. Therefore, we're gonna say U. U goes with A, and then G goes with the C. So the answer is this for this question and this for that question. Yes. Describe the process shown in the diagram above. Hey, what is this? This is translation. I told you guys. How many marks are this? Guys, don't tell me that you disappointed me. Unless you are not trusting me. Don't tell me that you disappointed me. There is someone who told me in the comment, I was so sad, and but I didn't stop. He told me that you lost me in the first 5 minutes. I knew that this one is looking for attention. Yeah, because I talk too much. Okay, I talk too much, but I wanted you to understand the concept. Yes. Anyway, what do we mark? We can have different ways how we can mark. But this one you are describing, name and describe. The process is called translation. That's one mark for the word naming. And then according to the messenger RNA, the transfer RNA with the complementary anticodon bring the required amino acid to the ribosome. The amino acid are joined together by the peptide bond to form a required protein. Simple. That's how I have been describing. But still in my book, the distinction material, I showed you exactly what you are supposed to do. Now the mouse is jamming. Okay, doesn't want me to this mouse doesn't want me to give you okay, let me use this. Okay, this mouse doesn't want me to give you answers, ne? Sorry. Yes, born it's there. Each transfer RNA is a marking point. Here is a marking point. This is a marking point. Each transferrin A carries a specific amino acid. We give you a tick there. When anticodon on the messenger matches the codon on transferrin A, we give you a tick. Specific amino acid, we give you a tick. Matches the anticodon. Yes. Then we are saying that this brings the required amino acid. Yes. Where does it go? To the ribosome. The amino acids are joined together or attached by peptide bond. Guys, we call them peptide bonds. We don't call them polypeptide bond. We call them peptide bond. I was very clear when I was explaining this. And then to form a required protein. That simple, short as that. But you can find a person writing a full page. So you're okay. That's short as that. That's what we wanted. Let's go. So this description thing, I'm not going to waste too much time because it is open thing, and you can find them in any book. Yes, you can find them in any book. Yes. Let's go to the next question. What are they saying? That's the moment. Yes. What are they saying? They're saying that the table below shows the codons. I explained this. Guys, take note of this because I explained it and I said this is one of the confusing subtopics. The table below shows the codons of codons that code for some amino acids. Okay, sharp. A mutation caused by DNA triplet to change this. Okay, so it means that we changed this to this. But they are saying this is DNA. But what is here is messenger. Meaning that to read this, I have to change it first. So I'm going to say that, okay, this was C. C goes with G. T goes with A. And then G goes with C. But changed it to C goes with G. T goes with A. And A goes with U. Because this is messenger RNA. Wabona? Yes. Now I come. I go to, you go to what? I go to this table. I look for it. GSC. 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 GSC is aspartic acid. What about this? Does it correspond to a different amino acid or the same amino acid? Let's go. And then you're saying that GAU, aspartic acid again. Wabona? Ntuli. I can call you Ntuli. Yes? Isn't it? I'm just saying Ntuli because Ntuli called me and said that. Thank you very much, sir, for teaching me. Yeah, I think I managed to pass. I pray for you guys, so that you pass. Sometimes when you're marking you're marking your students, and then you get something, you think of something, you end up giving a wrong one correct. You never know. Make sure that you pray until the results come back. Yes. Anyway, so since they code for the same amino acid, yes, so it means that the protein will not change. But these are 4 marks. Where am I going to get the 4 marks? You're gonna say that this change of this, yes, this changed into this. You writing it GAC changed to GAU. Those are marks. Those are 2 marks. This changed to this. Why add? Why this one is a mark and this is a mark? Is because you changed it. You see, you changed this. That's 1 mark, and you changed this. Those are 2 marks. And then this code for the same. This code is for aspartic acid as well as this code for aspartic acid. Meaning that these 2, they code for the same amino acid. Is another tick. Those are 3 marks. Then the 3rd mark, the 4th mark will say that this will not change the protein formed, or it will not change the functioning of the protein formed. Simple. Why? Because it's like calling you with the first name, calling you with the second name. You are the same person. Yes. Or sleeping on your bed when you are straight, and sleeping on your bed when you have carved somewhere, or does not mean that you're a different person. It's the same person. So they're just saying that, yes, however much the mutation occurred, but the mutation did not change the amino acid. Simple. This question, they have brought it back again. Tabulate two difference. I emphasized this question of mutation, and I told you that I know what students find when it comes to this question of mutation. Anyway, this question. Tabulate two differences between DNA nucleotide and RNA. I explained it before. Yes, those people who were on my channel yesterday. They are they are not asking you the differences between DNA and RNA. No. They are saying DNA nucleotide and RNA nucleotide. Clear? So there are two differences. The first one, DNA nucleotide, has thymine, and the RNA nucleotide has uracil. The second one, DNA is made up of, nucleotide is made up of deoxyribose sugar, and RNA nucleotide is made up of ribose sugar. There is no any other difference. They are looking for the nucleotide. They are not looking for DNA. They are not looking for they are not looking for just those nucleotide for the RNA and for DNA. That's all. So if you wrote a lot of things there, but there are 5 marks. Where is the fourth mark? Fourth mark is for the table. Just drawing a table and say DNA nucleotide, nucleotide. Even if you didn't write anything here, you draw a table, a complete table, you will get a mark. At least you are not getting 0. Yeah, at least. Yes. Let's go. Down syndrome. I talked about it also. Down syndrome. Okay, let's go to Down syndrome. What are they saying about Down syndrome? Okay, let's go. Down syndrome is the result of individual having extra chromosome on chromosome number 21. Okay. The genetic variation that can be caused by Down syndrome are, okay, trisomy 21, all somatic cells are individual, 3 copies of, okay, chromosome number, all. This word is a key point. And then they're saying that mosaic means something which is moving. Yeah, okay. Individuals have only some cells, not all. So this is a key point versus this extra chromosome, which is caused by abnormal process of cell division after. This is after. Is caused by after fertilization, but what occurs during gamete formation. This is during gamete formation. This is after fertilization. You see? Yeah. That's the major difference. The type of mutation lead to the trisomy 21. The type of mutation that leads. You can say chromosome aberration. Those who wrote chromosome aberration, correct? And then chromosomal mutation. Because these are chromosomes. Chromosomal mutation. Yes. Or aberration, correct. Abnormal process during gamete production that lead to 3 copies of abnormal process that lead, name the abnormal process that lead to the 3 copies of non-disjunction. The type of cell division that occurs after fertilization, it is mitosis. They're saying describe how the process mentioned above, which above B here, non-disjunction lead to trisomy 21 is 5 marks. This is a general question. Yes, go back to my distinction material so that we don't waste too much time there. Yes, I talked about it, but sometimes people they have challenge. But before I left meiosis, I said that please don't go without knowing this process, because this process they will ask you. Here is a question. Non-disjunction, failure of the chromosome to separate is they are looking for this, you see, this page. Each nucleus has, it contains 23 chromosomes after meiosis, but non-dissociation when occurs, okay. One nucleus will contain 22, while the other will contain 24. Yes, either of the resulting gamete will fuse with the normal gamete, but they said, yes, with the normal gamete. They said, yes, they said what will happen. Yes, so the normal gamete is 23, then the resulting, the 20, if the 24 fuses with 23, it's gonna be 47. The 22 fuses with the 23, it's gonna be 45. Yes, so in this case they are looking for 23 because they're saying trisomy 21. It means that the 21, it has an extra chromosome, not short of a chromosome. Yes, let me explain to you. Yes, and then they are saying that, let me show you what they are trying to say here. Yes, and remember, during non-disjunction, this is a cell, ne? Under metaphase. Yes, so this is what happens. So during non-disjunction, the whole set is going to go to one side, so meaning that I'm going to have 2. So the normal one is going to be 2 cells. I'm going to have 2 here and 2 here. But during non-disjunction, the whole of it is pulled to one side. And then this one, yes, it will go to one side. And then this one, another one will go to one side. Meaning that here, yes, here I will have 1, 2, 1, 2, plus this one, 3. And then this side, I'm going to have only 1. So this one has extra chromosome, 2, 3 instead of 2. And then here it has short. So they are looking for this because they are saying, yes, if it occurs on chromosome number 21, it becomes trisomy 21. Tri means 3, 1, 2, 3. You understand? So now when this one fertilizes a normal ovum, then it's going to have, now is the cell is supposed to have 23 chromosome. But this one is not going to have 23 chromosome. It's going to have 24. So if this fertilizes a normal ovum, which is 23, it's going to result in 47 instead of 46. So that's what they are saying here. So go to the distinction material. Yes, get there. So that's what they are trying to explain, or they're asking you here in this question. Let's go to the next question so that, yes, this describe 2 differences between trisomy 21 and the mosaic Down syndrome. Yeah, I think I talked about it. Trisomy 21, all cell, check, all somatic cells in the individual have 3 copies of trisomy of chromosome 21, you see, all. But here, yes, make sure you draw a table, ne? I think they don't want a table. They're not saying tabulate. While here in mosaic, the individual has only some cells with extra chromosome 21, but here all the somatic cells. The number 2, the process, when does it occur? It occurs during gamete formation, while here it occurs during or after fertilization. Those are the 2 differences they are looking for. It's just as something from there which you can use to get the answer. All right, I see ourselves trying to finish soon, eh? Yeah, so we are going to the question number 3. Question number 3. How is the paper? Let me know. How is the paper? Let's go to the next question. Ne? Yeah, I think the paper was simple. Ne? The paper was simple for those who read. Yeah, let me say like that. And I think I should come back and I teach you how to read. It does not stop only here, even when you go to the university. Good. All right. The diagram below shows the inheritance of tithe. Oh, they like this funny name. Anyway, I'm not going to remove my teeth because of these names. Okay. Is a rare disease which lead to this destruction of the neuron. It is inherited as Autosomal. Okay. Autosomal means that it occurs in the body cells, meaning that it's non-sex linked. It's occurring in these other cells, these other chromosomes, not on chromosome number 23. Yes. Okay. TT. So meaning that you must not use X and Y. This is not sex linked. It's autosomal disorder. Anyway, let's go. Describe what is meant by is a disorder which is caused by a gene carried on non-sex chromosomes. Yes, non-sex chromosomes, meaning that from chromosome pair number 1 to chromosome pair number 22, it must not be sex. Yes. How many sons do individual 1 and 2 have? 1 and 2. Yes, sons. Son is a box. It's 1, is 2. But this son is not belonging to these two. You understand? This guy, yes, just married the daughter of these two guys, meaning that the answer is 1. You understand? You see? Yes, this is just an intruder who came to marry the daughter. Yeah, the beautiful daughter of 1 and 2. Yes. So the answer here is 1. Using the individual 3, 4, 5, explain why it can be concluded that this disorder is controlled by a recessive allele. 1 is 3, 4, 7. 3, 4, 7. Okay, let me clear up here so that at least, yes. Okay, let's see. 3. Okay, this is the affected individual, né? And these two people who are, if they produce a person who is affected, it means that these two people were heterozygous. Meaning that it is capital T, small t, capital T, small t. You see? Since they carried that what? Since they carried that gene, this gene, recessive allele. And then this recessive allele and this recessive allele gave rise to this. Meaning that, what is the question? What are they saying? Which question is this? 2.5.3. They're saying that it means that individual 3 and individual 4, they are both normal individuals. I see. That's the first mark. But they gave rise to individual 7 who is affected with the disease. You have to tell us the disease, né? A female who is affected with the disease. Yes, that's the second mark. This means that individual 3 and 4, they were heterozygous. Meaning that they are heterozygous because they are both normal, but they gave someone who is sick. Meaning that they were heterozygous, they were carriers. That's another mark. And then they pass their recessive allele. 1 recessive allele from 3 and 1 recessive allele from 4 to individual 7. That's another mark. You're able to get all the 5 marks. 1 recessive allele from 3, 1 mark. 1 recessive allele from 4, another mark. Yes, to form this one who is affected with the disease. So, yeah, it is an application question, whereby you have to apply and explain exactly what happened to inform individual 7. Then they are saying that individual 1 and 2 can produce children with 3 possible genotypes. Okay, list those 3. Individual 1 and 2 can produce, yes, individual 1 and 2 is the same thing. 3 and 4, they are both heterozygous. Okay, they can produce, if they are both heterozygous, it means that, look, it means that this is capital T, small t, heterozygous, capital T, small t. So it means that when they produce, they're going to produce capital T, capital T. This is what they are trying to show you. Capital T, small t. This is capital T, small t. And then small t, small t. You check these two. They are both heterozygous. This is homozygous and this is a homozygous. Let's go. The question. They're saying that list all the possible genotypes that have 25% chance of offspring. Then this is 50 and this is 25, this is 25, meaning that I'm going to come here and say capital T, small t, a comma. That's all. That's what they're looking for. They give you ticks. In human, hemophilia is caused by a recessive allele of X. Of X. Anyway, it's caused by a recessive allele of X. Let's go. Of X. They're saying that in humans, hemophilia is caused by a recessive allele of X. Okay. This is carried on X chromosome, meaning that this is sex-linked. The moment you say that, that's sex-linked. Hemophilia, yes, we know is a sex-linked disorder. A woman who is heterozygous for hemophilia marries a man who is heterozygous for hemophilia. That's a woman. It means that it's like this. Capital H, small h. Yes. Marries a man who is hemophilic. Yes, meaning that the man is definitely is like that. Is use the genetic cross to show the percentage chance of couple having having a daughter who is homozygous for normal. Homozygous for normal and is a daughter, meaning that this person is gonna be like this. There's no way. Check, the father is not normal. Then it means that the answer is gonna be 0. 0. Yes, but they are saying use the genetic cross to show the percentage chance. The percentage chance is 0% because there is no way. But they are saying use the genetic cross to show. So you're saying 0% is 1 mark, but you have to show. Don't forget to show all the steps in a genetic cross. Let me see, is it P1? Let me see. A man who is, they didn't talk about anything. So I'm going to come here and say P1, F1. P1, parent, phenotype. You go to the question, you'll get the phenotypes there. I told you that this one is simple, and a man who is heterozygous, a woman who is heterozygous, meaning that this is a normal woman. Yes, marries a man who is hemophilic and a man who is affected. Then you go to genotype. Genotype, I'm just rushing so that a lady was like this because they say that heterozygous, marrying a man who is like this. Yalla, let's go. They're saying like that. Don't forget that these are gametes. I always tell you that they are not at the beach, so they must be circled. If you didn't circle, it's still fine. Don't stress. Give you a tick. These are gametes. Wabona. Yeah, but how do you call this process? Meiosis. You saying P1, F1 will give you a tick. So that's the second tick. 0% is a tick. But it's the second tick. This is a third tick because we got it. So you have 4 marks. Mahala. The question is out of how many? 7. So we are searching for the 3. This is the next tick. It's the next tick. Then after that, you can draw your Punnett square here, or you can just cross. When you cross, you'll find out that this, look critically. This goes with this. So it's going to be X, capital H, X, small h. Then this goes with this, capital H, small, capital H, yes. And then this with this, small h. This with this, small h, and then y. Is there any probability of having a child who is homozygous normal and is a female? No. There is no way. Therefore, this is offsprings. F1 offsprings. And then you remember you have to cross, show cross or the Punnett square. So, and this process is called fertilization. So we give you another mark for this. But what I know that the marks are more than what we stipulate. But you saying that therefore the percentage chance is 0%. This is going to be a compulsory mark because the question is emphasizing that percentage chance. They want a percentage chance. You understand? Yes. So it's 0%. Having a child who is normal, homozygous, and is a female. Yeah. Let's go to the next question. I think I'm clear. Yes. Let's go to the next question. These papers are simple if you read. If you don't read, yeah, they become sour. But I don't expect you to. Let's go to the next question. The next question. Question number 3. It means that now you have 100 marks. 100 marks. See now, you are approaching slowly a distinction. You're approaching a distinction. I know some of you just made small mistakes, which can be acceptable, but there are some mistakes which cannot be accepted. So, yeah. Anyway, let's go. Yeah, let's go. This is year 2023. Yeah, I know some of you gonna be successful. I pray for you so that you become successful. Anyway, learners conducted an investigation to determine the blood group was the most common in the community. I talked about a pie chart yesterday. Yeah, they conducted information about, okay, 200. It's a big number. It means that the sample size. Take note of that. In each of the 3 types of blood group. Okay. Yalla. Let's go. They're saying that in their community, they did not include the first donor in the investigation. Why? Comparison of results. Okay. Yeah. The pie chart below shows. All right. Let's go. The percentage of each blood group in the sample. I know you read all these questions. You know them. So I'm not going through too much. State the aim of the investigation. Ooh. The aim must start with the word to and is always put in the first statement or second statement. That's why always I tell my students. If you can't find it, then you have to figure it out. Learners conducted an investigation to. That is the aim. From to, the investigation. The aim is to determine which blood group was most common in their community. Yes, in the learners' community. Conduct an investigation. Yes. So the aim is done. Answer the following question. State 3 planning. We always ask you this, but you go back and read the question and see the possibilities for you. What are you going to plan? What are you going to do? So, but I need these people who are going to be giving me blood. So you have to organize them. Yes, get those 200 people. Yes, you are not just going to just get them. Yes, you have to get permission because this is a community thing. Get permission where you're going to do this, or the clinic, or try to go inform them so that they know and they accept. You are not going to force them. Get permission so that they can do that. You are not going to just collect the blood just like that. Get the necessary equipment, planning before you do the activity. Yeah, that's what they are looking at. Yes, something like that. State one way in which learners ensured. Yesterday I told you the word language and I emphasized it. If they ask you, don't ask you, how did this person ensure, how did the person do or increase reliability? Or how do you ensure reliability? They say that, how did the learner ensured? That is a past tense. Your answer must be in a past tense. The learner ensured reliability by using two. Remember, reliability is to increase the sample size by using a big sample size by using 200 people. 200 people. You see? So, this learner used the 200 people. Sharp. So, that is done. Give the reason why give one reason why they did not include the, yes, we said comparing the result. Calculate the percentage, the number of participants that had blood group B. Show your working. Blood group B. They use 200, but only 15% blood group B. So you just come here and say 15 out of 100 times 200. So the answer you get is the answer, around 30%. Done. They give you your 3 marks. Give you a mark, give you a mark, give you a tick. Done. They're saying that name the blood group that has only recessive allele. Only recessive allele, blood group O. Remember, it is recessive allele. Remember? Yes, I said it yesterday. Results of codominance is AB. I said it yesterday. Remember, it's A and B. Give the possible genotype of the blood group represented by 25%. 25%. Where's 25%? Is A. But they're saying the possible genotypes. If you remember, A is either homozygous. Go to the distinction material. You will see what I was saying yesterday. A heterozygous. I explained this yesterday. I explained it very well. Very well. Yes. I hope you guys did not disappoint me. Ne? I don't want the marks to come back and then I'm disappointed. Say, oh, why did I do this for my learners, these learners of mine? Ne? No. I don't want that. Ne? I want everyone to pass. Ne? Yeah. I want everybody to pass. Anyway, let's go. Question 3.2. We are almost done. We are almost done. Anyway, question 3.2. These are dogs, man. Do you like dogs? Let me know the question in the comment below. Do you like dogs? Do you have a dog at home? Sometimes they call you a dog. And a dog. Okay. Dogs have been selected and bred over many years to produce approximately 340 different dogs breeds. They have been bred for certain characteristics desirable to humans. Saying that analysis of 736 okay, gray wolves, only direct ancestor, present day dogs bred. Okay, all the dogs belong to, I'm just reading so that I get the idea, ne? But you know, you can read it for yourself. Okay, I've got the idea, ne? Very fast. They're saying that name the process whereby the different breeds of dogs were produced. Artificial selection. Yes, these are the things we are talking about here. Where am I? Yes, artificial selection. Is it? Yes. Where is it? Okay, this is meiosis, ne? Yeah, artificial selection is they are looking for you. These dogs, even this question has these dogs we are talking about. Exactly the same dogs. Don't disappoint me. Don't disappoint me. Don't disappoint me, guys. Dogs, wolves. They're there. Born. Check. They are dogs, the wolves. That's what are the wolves we are talking about. Okay, they're saying that. Name the artist. What did we say? Is what? Artificial selection. Human, they select desirable traits. You see? That's what they said here. Desirable. They're desirable to humans. Yeah. Yes. Distinction material. Get it. Yeah. Get it. I'm talking about those who, your sisters, your brothers. Yes. Anyway, let's go. And we're saying that name the process whereby different breed of dogs were produced. Artificial selection. Describe how human carried out the process named above. Meaning that they got the characteristics they wanted. They let that individual which has that characteristic to mate with the other characteristics they want, and then the other one which doesn't have the characteristics were not allowed to give to have sex, meaning that to pass their genes to the next generation. So after many generations, they had to be with that desirable characteristic. That's what they are saying. So if you look at this example, check, it was about milk. Choose or select the cow in your herd, or now they're talking about the dogs to produce which has that characteristic. Yes. Then they are saying that let only those dogs to reproduce, the one which have the specific characteristics, then select the offspring that produce that characteristic most. Yes, let those only those offspring reproduce, allow them to have sex and have offspring like that. Keep repeating this process of selection and then the breeding until you achieve your goal. That's it, to get the desirable dog you want. Explain how it can be confirmed that all the dogs breeds belong to the same species. Same species. It means that they can interbreed to produce a fertile offspring. If they are not producing fertile offspring, then they don't belong to the same species. Yes. They must produce to produce a fertile offspring. Hence, this answer becomes right. Yes. Explain how present-day dogs may have been disadvantaged. Yes, in the relation to their common ancestor. This common ancestor wolf, Baba, it could resist diseases. Now, you have to take it to the how do they call it, the veterinary doctor. They were staying in the wild, so these dogs are susceptible to diseases. Less chance of these dogs being adapted to the environment. Yes, these dogs, the one which are new, yes, they have less chance. This one, it could adapt, Baba. Yes, meaning that some genes are being lost as you are doing this selection. Yes, so the dogs are being disadvantaged. Yes, let's go to the last question. Describe the process of speciation by geographical geographical barrier. Guys, I told you don't go to the paper. Speciation is here. This is what they're looking exactly. This formation of new species. Exactly. There is a population of a particular species. A population of a particular species becomes split into two by geographical barrier. As a result of, you don't talk about geographical barrier. Okay, you can say river, mountain, or whatever, but geographical barrier. As a result of two parts of the population cannot interbreed, there is no gene flow, yes, between the two. The natural selection will occur independently, yes, due to different environmental conditions. As a result, the population became genotypical. You start with genotypically, then phenotypically different over time. Even if you remove you bring these two populations, you mix them together, meaning that you remove the barrier and you let them mix and they interbreed, they cannot produce a fertile offspring. They cannot produce a fertile offspring. Therefore, one or both population have become new species. This is exactly without changing any word or what. That's what you're supposed to write. That's what you are supposed to write. Someone is gonna tell me that what if I used a scenario? That's not KO because they didn't give you a scenario. KO is when the examiner sees, just give you a tick, not arguing with him or herself. Yeah, should I give no? Something like that. Don't like it. Just when examiner sees your paper, is marking it as you are. You know, when you're marking someone, you see that someone is getting all the ticks. Yeah. You feel good, Baba. You feel good. You feel good. The same way you feel good when your marks are coming back and you have distinction. How do you feel? You feel like you are not fitting on the when marks are nice. All right, let's go, man. You feel good the same way I'm feeling good when I'm teaching you. Yeah, I feel good, ne? I feel good. All right, let's go. They're saying that. Name the type of diagram represented above. Okay, the diagram represents the evolution of the family Hominidae. Okay, sharp. They're saying that. Name the diagram. I showed you these things. Phylogenetic tree, and I showed you the questions you're supposed to answer, and I don't think that they skipped those questions here. Let's go. Name the diagram shown above. Phylogenetic tree. Done. Give the letter of organisms, which is the common ancestor for all the hominids. P, because all of them, they come from here. Is P. They're saying that. They're saying that. What is common? Okay, share most recent common ancestor with the gorilla. Gorilla is here. Gorilla is there, but they share a common ancestor. What shares the common ancestor is here, meaning that is this. But what shared is R, because gorilla and R, they share a common ancestor. So the answer here is gonna be R. Give two organisms that have S as a ancestor. Two organisms that have S. Where's S now? S is this. Is this point. Stop it. Stop it when technology. Okay, this is S. So meaning that you're gonna say this and this. Those are the two individuals. Let's go. Name two organisms that have S as a common ancestor. We have said it. Quadrupedal. These ones. Gorillas. You know them. Is chimpanzee quadrupedal? I think so sometimes. But these ones, they are quadrupedals. Okay? They're saying that describe three anatomical features of the skeleton of a quadrupedal organisms. Skeleton, when you talk about a skeleton, you're gonna talk about the spine, the cranium, because that's a skeleton, the spine, the cranium, and then the pelvic girdle. Yes, talked about. I showed you here when talking about. Yeah, for the case of the skeleton, here, see, it's a skeleton they're talking about. They're saying about quadrupedal organisms. Foramen magnum is in forward position. Yes, it's a skeleton. Yeah, that. Yes. You talk about the spine is C-shaped, and then long and narrow pelvis. That's what they are looking for. Simple. Simple. As simple as eating ice cream. No bones. Yeah, let's go. Second last. Yeah. Fossil evidence is used to support out of Africa hypothesis. State out of Africa hypothesis. Modern humans originated from Africa and migrated the rest of the world. I told you when I was concluding, don't go to the paper without knowing this. Modern humans originated from Africa and migrated the rest of the world. And I showed you this. That take note of this. Unless you do not want to trust me. The modern humans originated from Africa and migrated the rest of the world. That's what they want. The word modern must be there. Modern. Don't just say humans migrated the rest from Africa. And Homo habilis, Homo naledi, Homo neanderthalensis, all those are humans. But the modern ones, Homo sapiens. Yes, they're looking for. Okay, let's go. Last question. Describe how the fossil evidence is used to support out of Africa hypothesis. How is this fossil evidence? How is this fossil evidence used to show out of Africa hypothesis, guys? How is it used? Describe how this fossil evidence is used to support out of Africa hypothesis. You guys, I told you this and then you didn't write it. Okay. You talk about all the Adipithecus, Australopithecus, Homo habilis. You say that only, the word only must be there. Only. Adipithecus, fossils of Adipithecus, fossils of Australopithecus. You can talk about the examples. Fossils of Homo habilis. Only found in Africa. Then when you go to Homo erectus, you talk about the oldest fossil of Homo erectus, distinction material, and the Homo sapien. Homo erectus and Homo sapiens. Here you talk about oldest fossil of Homo erectus and Homo sapiens were found in Africa. Don't say that only found in Africa. Nope. You have to say oldest. The key point there is oldest. That's why I put it in bold italic and I was supposed to underline it so that your eyes can open when you are reading it. I think, guys, the paper was easy. Yeah. Don't forget to subscribe, like, and share. It has been M.SAIDI, has been taking you all over this paper. I think, yeah, the paper is a little bit easy. It was a little bit easy. Okay, it had some challenging questions, but it was not that bad. It was not that bad. Yes. Thank you very much for being with us. Yeah. Thank you very much, guys. It has been a wonderful paper. Don't you think so? Let me know in the comment below to know how was this paper to you, based on you. How was the paper? Yeah, let me know in the question in the comment. Thank you for being with me from the beginning until guys, ne? Yeah, I just love you so much. Stay blessed, ne? Stay blessed, ne? Yeah. Maybe I just a small note that even physical science, yeah, even other papers we will be doing discussing them immediately after you have done them. Thank you very much, ne? See you.